So after we have learned in the previous lecture about the mechanism, molecular machinery that's involved in vesicle fusion, and now we're going to talk more about different ways that vesicles actually can fuse with the membrane. So we have learned last time that its near complex is going, together with the synaptotagmin, is going to bring those two membranes in a position. Now we're going to learn what it can lead to and how are those vesicles going to be recycled, and about all different methods that we can use tools to study and to dissociate between these different ways. So this is the part that we came to the fusion pore formations. Near complex together with synaptotagmin is going to lead to the fusion of two membranes. Now what can at this point happen? We can either have the fusion pore expansion and release full fusion with the membrane and full release of neurotransmitter, or this can be temporary and lead to the closing of the membrane, leading to kiss and run. There's a third way as well. This can lead to the opening, closing, and then again opening, which would be opening then plateauing. So there are different mechanisms by which neurotransmitters can be released, but the two main different mechanisms are through kiss and run, in which again fusion pore is open but then closes, and then, or we can have a full fusion in which full amount of neurotransmitter is released. It is not really well understood when and how cells decide between kiss and run versus full fusion. So this is a matter still of a lot of exploration when cells decide to go kiss and run and when to decide to full fusion. But you can appreciate that there is going to be a big difference in response of postsynaptic cells, because you will release different amount of neurotransmitter in the synaptic cleft whether you decide to do kiss and run and full fusion. Now, what are the experimental approaches with which we can measure whether it's kiss and run and full fusion? So there are both optical and electrophysiological methods to study. So here are examples of some optical methods. They are dyes. FM dyes are lipophilic dyes that are membrane impermanent dyes that will stay at the active terminals. And then you can stimulate cells to have endocytic uptake of vesicles. So all of the vesicles are going to be labeled with that dye. And then when you induce the fusion, then that dye is going to leave. So for example, if it's kiss and run, you're going to have some release of the dye, but then you're going to go back. So here, if you are doing the imaging, you're going to see the decrease in fluorescence that's then going to stop. On the other hand, if you have a full collapse fusion, this release is going to be much deeper. So you're going to have much quicker and complete decrease in the fluorescence. On the other hand, you can have genetically encoded fluorine-based probes. What is fluorine? It's a GFP-based pH sensor, which is dim and low pH. Now, synaptic vesicles have very low pH, and we're going to learn that in the next lecture because that's required to fill them with neurotransmitters. But what you need to remember at this point that they are very low pH, so it's dim, and then when the synaptic vesicles are open, they lose this pH. The pH increases and leads to fluorescence increase. You can fuse fluorine to different molecules that are present in synaptic vesicles. Here are examples of synaptobrevin that we talked about. It's really important vesicle fusion. You can fuse it to V-GLUT1, which is a transporter that leads to transport of glutamate inside of the vesicle, or synaptophysin, which is present in a huge amount of vesicles. Anyway, you can use any of these proteins. You can also use the things that are secreted, such as BDNF. But pretty much it's going to be dim, so there will be no fluorescence, and then the opening of the pore is going to lead to increase in this fluorescence. But if it's a kiss and run and you have very quick closing of this pore, then you will very quickly lose this fluorescence. On the other hand, if you have a full fusion, this is going to lead to increase in fluorescence. It's going to be maintained as long as this vesicle is fused with the membrane, and then it's going to decrease back. Remember, it decreases back, and because this does not leak out of the vesicle, these are associated with the vesicles and they will stay. In case if you label with pH fluorine, for example, BDNF, that's going to peptide, that's going to be released from the vesicles, then in that case you will completely lose it. It's not going to be retrieved. You cannot re-image it afterwards. Third method that we have here are use of quantum dots. So these are single nanoparticles. They're bright, and again, they're around 15 nanometers big, so they can fit in the vesicle, around usually 1. or two, and they are also having pH dependent fluorescence. Again, it's going to increase and we lose the low pH that's normally present. So here is a short opening, leads to transient increase, and then it goes back. On the other hand, if we have full fusion, it's going to escape. So now this is a short increase followed by a huge decrease because we do not have even the dim nanoparticle, dim fluorescence that would be normally retained if they stay in the vesicles. So these are optical methods. They allow you to study them and the synapses. So these give you the space. As you can see, the time is not extremely fast. This is not as fast as a synaptic transmission. In order to have that resolution, you can use electrophysiological methods. You can measure whole cell capacitance. They reflect increase in the membrane surface area increase upon fusion. When vesicle fuses with the membrane, it's going to increase the membrane surface. Now, those are much more useful for the measuring of diffusion when we have the secretory cells, when they have huge synaptic vesicles. This is a little bit more difficult to do in neurons, which have smaller vesicles. But pretty much release is going to lead to a short increase in the membrane surface. So if it's kiss and run, it's going to be really temporary. And now keep in mind, we have now in a millisecond range. On the other hand, if it's a full fusion, you're going to have a little bit longer increase in the capacitance in this case, and as long as the vesicle is not then again removed from the membrane. Another really good method to use is amperometry. Amperometry is using the original use was using carbon fibers, which can actually oxidize dopamine or serotonin, and that way it make electrons, and you measure the current. So you can actually measure the amount of neurotransmitter released at the synaptic space. So again, resolution will depend how thin your carbon fiber is. And now these are modified, so you can measure also glutamate, because they put enzyme on top of those fiber, which can then convert glutamate to the metabolite that actually will be able to be oxidizable molecule. And again, if you have, in case of kiss and run, you're going to have a temporary and a smaller increase, while in the case of full fusion, you will usually have this shorter foot and then higher increase. And this is in case was serotonin measure here, so you have much bigger release. And keep in mind that there are different scales here, 0.25, 0.75, so this is a much bigger increase than we have with the kiss and run. Finally, what we have talked before, you can measure the postsynaptic currents, which has been what people have been measured from before, either changes in membrane potential, or in this case, you can measure AMPA and NMDA receptor currents to be the measure of how much of the neurotransmitter have been released. So in the case of kiss and run, you're going to have a shorter or a smaller amplitude of these currents, while in the case of full fusion, it's going to be bigger. So therefore, the amount of neurotransmitter release you can see here directly will, whether it's through the kiss and run or through the full fusion, will directly influence the postsynaptic response. Now, in order to maintain this rapid signaling in the CNS, you may have to have a lot of synaptic vesicles that have to release their fusion, that have to fuse with and fuse with the membrane and release the neurotransmitters. So what we know now, using these methods that we just talked, that there are actually different pools of synaptic vesicles. In general, as accepted, there are 3 different pools, and you may find different publications which they give them different names or categorizing different number of pools, but in general we consider 3 different pools. One, a readily releasable pool, which will consist only of a few vesicles. It's around 1%. So these are the ones that are docked and primed to release. This is where the snare complex is already formed, and synaptic tegment is just waiting for the calcium to come and fuse with the membrane. So they're just a small percentage, and they are the ones that are going to be immediately used. So they're docked, they're released very quickly, and they're very quickly recycled. Then we have a recycling pool of the vesicles, which is a bigger percentage, 10 to 20%. They are not docked, they're very close, they're scattered, but they're very close, and they're going to be released within a few seconds. So they will be moving a lot because they're not docked, but again, these are not the next level. So if we measure, this is an example, for example, when they use that FM dye. So with readily releasable pool of the vesicles, it's very quickly released, so it's almost instantaneous. Then it takes a little bit longer to, when you keep constantly stimulating those neurons, presynaptic neurons, to release recycling pool, and it takes much more stimulation and much longer time to release reserve pool, which is thought to be 80 to 90%. It's actually thought this reserve pool may not really be used in a lot of physiological situations. However, these are the two pools, again, recycling pool and readily releasable pool, that are used a lot, and they are going to be recycled in order to maintain ability of synaptic transmission. So how are they recycled? So the first evidence for recycling came actually not from the methods that we talked about, which you can now do much better. This was in 1973, they didn't use these FN dyes as much. So what they used was the enzyme horseradish peroxidase that they would put in a bath solution. They would stimulate release of the vesicles, they will stimulate presynaptic terminal, and then this enzyme will also be picked up in the vesicles. And then they would wash away HRP, and they would wait for a while, and then they would stain, and they would see that these synaptic vesicles contain HRP, which would suggest that they were formed when HRP was on the outside. So the idea was that this goes through the clathrin-mediated endocytosis. So we have docking, priming, and fusion, and then they have budding through the clathrin-mediating endocytosis that happens around 10 to 20 seconds. What we now know is that there are actually 4 different mechanisms of recycling of the vesicles. First one is if you have kiss and run method of fusion. You can have also kiss and stay, which are very similar. In kiss and stay, it actually does not move from being docked. Kiss and run, it goes into this readily recyclable pool, so it very quickly gets recycled. So the pore opens, and then immediately it can be used to be recycled. Then you have a full collapse. In case of full collapse, there's 3 different ways by which those vesicles can be recycled. One is going to be through the clathrin-coated pits, and we are going to talk a little bit more about it because there is most evidence about it. Then there are 2 different ways of endocytosis. For a long time, we knew about the bulk endocytosis. That does not happen at the active site, and you can retrieve a huge amounts of membrane. Now we also know there's something called ultra-fast endocytosis, which is pretty much a sped-up version of bulk endocytosis. So it functions in a shorter time scales, and is thought to be able again to retrieve a huge number of vesicles when there is a high frequency stimulation. I have included this one as well because it might not be as evidenced in this scheme that the kiss and run or kiss and stay, depending on which of the mechanism, really occur at the active zones, and then the other mechanism of clathrin coated and the bulk endocytosis, whether it's going to be ultra fast or regular bulk endocytosis, do occur on the periphery on the synaptic of the active zone. And again, these different mechanisms of vesicle recycles with different how long it takes. So kiss and run and kiss and stay are really quick. This happens within less than 1 second. Clathrin-coated pits are 15 seconds to 1 minute. So it's a little bit short. Bulk endocytosis is the slowest one, but the ultra-fast again can be in the milliseconds. So what is the molecule? Again, the clathrin-mediated endocytosis is the best studied one, and we know most about it, and there's most evidence. It uses clathrin. This is a typical endocytosis mechanism. So it uses clathrin triskelion. This is a very interesting molecule that forms these clathrin coats. So what happens is, first, if this was your fully fused vesicles, first you will have adapter proteins that are going to recognize proteins that are on the vesicular membrane. For example, we know that synaptotagmin is one of the molecules which is recognized by adapter protein called adapter protein 20. Now these adapter proteins are going to bind to proteins that are in your synaptic vesicles and to the clathrin. And clathrin is going to induce this curvature of the membrane. Also, adapter proteases are going to be helping with inducing the curvature of the membrane, which is really necessary to make this budding of or endocytosis of the vesicles. Then, after they assembled into this coat, the curving membrane. Then we have dynamine, which is kind of a scissor molecule that comes and pinches the vesicles outside of the membrane. So now they are endocytosed, and what we now have to do is we need to remove all these cluttery molecules. So we have HSC17 that help remove all these extra proteins so that the synaptic vesicles then can be filled with neurotransmitter and recycled. So we have measured, so this is something I want you to work at home. We talked that transmitter release can be measured by interferometry. Remember, this is a carbon fiber. And again, this is still a reaction that occurs, so it can be oxidized, and you can measure the current. So this is example of release, and then presumably you can put it right next there to sense the amount of neurotransmitter release. And this is showing you again how it can show you whether it's a full fusion or just a kiss and run fusion pore alone. So in complete fusion, you may have a foot where you just release a little bit until completely fuses, and then complete fusion gives much longer release, and then fusion pore alone, you just have this little short ended foot. But the bottom line of this is you can actually get a quantitative measure of how much of neurotransmitter is released. And this is the method that has been used first time to actually quantify the quanta, or quantify the number of neurotransmitter vesicles that are present in one vesicle. So that's the problem I want you to think at home. You will be using a parameter to measure the quanta or amount of dopamine or serotonin released during single vesicle fusion. So this is on the left, this curve that's obtained with the known amounts of serotonin that confirm that oxidation produces 4 electrons every time. So this was, again, what was experimentally gotten, and then the best fit that says it's going to be 4 electrons. On the right, you have the histogram that shows you the charge measurement with a single vesicle release. So it shows you how much of charge is going to be released with a single vesicle. So this is around 60 something. So using the formula Q equals M multiplied by E and multiplied by Z, where Q is the charge, which you have here, M is number of the molecules, and E is elementary charge, which is here present, and Z is the number of moles of electrons transferred per molecule of serotonin, which is actually 4, you know, from this diagram. I want you to calculate the average number of serotonin molecules, so this would be a big M. So you can see what is the most frequent charge that you get here, and then you can, based on that, E, Z, which is 4, calculate the M, which is number of molecules that is released per single vesicle.